next chapter is about cell communication and how cells communicate with each other. So once again, I have put a bunch of different learning objectives and essential knowledge that you're supposed to have in regards to the College Board. So there you go. As you can see, this is a relatively important chapter because there are a lot of pieces associated with it. So basically, if you're a multicellular organism, it's really important that your cells be able to communicate with one another. So biologists have discovered some mechanisms of cellular regulation that are the same between different species and different organisms. Most often, cells are going to communicate with each other using chemical signals, like, for example, hormones, like the fight or flight response. And here is an example. So this gazelle is probably pumped up on adrenaline and running away. I'm sure the cheetah is also experiencing some hormonal changes that will also improve his ability to run and hopefully catch this gazelle. Well, hopefully if you're the cheetah. I guess it's not so good for you if you're the gazelle. So a signal transduction pathway is the term that we use to refer to a series of steps. And a signaling molecule will attach to some type of protein on the cell's surface, and then it's converted into a response within the cell. So what this pathway does is it takes a molecule outside the cell and basically turns it into a message, and then the cell will respond from that. So there are similarities between pathways that suggest that these signaling molecules evolved in prokaryotes and then later on were modified in eukaryotes, which is kind of a hint at the evolution of life on Earth again. So there's a really awesome video that I have posted on your Schoology. It's a TED video about bacteria talking to each other. So please make sure you check that out, the TED video on Schoology. And it has to do with the signaling molecules and bacteria sensing local population density. So here's a different example, though, different from the one that's on that video. This yeast, I'm not quite sure how to pronounce that, but it has two mating types. It has a type A and a type alpha. So this type A, or the purple is the type A and the pink is the type alpha. And cells that are of different mating types can locate each other by secreting factors that are specific to each type. So as you see here, an alpha yeast cell secretes a pink factor, which then binds to the receptor on the surface of the A-type cell, and the A-type cell does the same. So you can see the shape of the signal matches the shape of the receptor. There's a specificity here, similar to what we experience with hormones. So here is a type of soil-dwelling bacteria, and they use chemical information to share information about what nutrients are available. So here is the bacteria, and here the bacteria is signaling to aggregate or clump together, and over here it forms a spore structure, which is sometimes also referred to as a fruiting body, which looks like this on the surface of something. And it will do this when nutrients are available. Okay, so cells can signal locally and long distance. So when we're talking local, we're talking about, with the cells around it. And when we're talking long distance, we're talking without or throughout an organism. So if you were a plant cell, plasmodesmata allows you to directly exchange chemical messengers between two adjacent plant cells. And there are different types of junctions between animal cells, and one of them is called a gap junction, and that will allow direct exchange of chemical messengers. And then we also have local regulators, and local regulators can be secreted by glands or cells of glands or something like that. And they're only going to travel short distances. Then there are long distance signaling mechanisms that have receptors on the surface of the cell, and those allow signals that fit specifically with the receptor to be sent long distance throughout an organism. So this right here is showing you the gap junctions. You can see the spaces here between the cell membranes as these two cells push against each other, so molecules can be exchanged directly. And the same thing with plasmodesmata. The plasmodesmata are holes in the cell wall. And here we have cell-to-cell recognition. So there's a receptor here. Here is the receptor, and here is the identifying molecule that fits with it. So the cell might, trigger some type of reaction in this cell. And then we have our long-distance signaling over here. So some examples of that are more like hormones that travel within your bloodstream to cells that have receptors that will specifically bind with those hormones. So there are 3 stages of cell signaling, and Earl Sutherland discovered kind of these stages by learning about epinephrine. And his work suggested that cells go through 3 processes. They have a reception, a transduction, and a response. So reception, remember the root of that word is to receive, so the receptor will receive the signal as it's doing here in 1. Transduction is where the signal gets relayed through different molecules until finally there is a cellular response, and that is the third part. So in reception, the signal molecule is always going to be called a ligand. This is a very important term, so make sure you commit it to memory. The ligand and the receptor are highly specific. Usually, the ligand will cause some kind of shape change in the receptor, and that is the initial transduction of the signal, so the first part of the signal. And most signal receptors are going to be plasma membrane proteins. So when the signal is water soluble, then most likely it will bind to specific sites on a receptor protein that spans the plasma membrane. And an example of this are G protein coupled receptors. So a G protein coupled receptor, or GPCR, those are the largest family of cell surface receptors, and it's a plasma membrane receptor that works with the help of the G protein. And the G protein is going to act as an on-off switch. So if GDP is bound to the G protein, then the G protein is inactive. So, and this DP relationship is still diphosphate. So if we look over here, we have our G protein coupled receptor in picture number 1, and we have a G protein that has GDP bound to it here. So this G protein is inactive. However, if a signaling molecule or a ligand comes in and activates the receptor, the GDP then loses, or it uses energy here, this G protein right here, uses the energy from GTP becoming GDP to move along the membrane as it does over here in the third picture, activating this enzyme right here, which will then cause some type of cellular response. And then an inorganic phosphate here will come back along and reactivate once necessary. This is a computer rendition here of both your plasma membranes and the types of receptors that you might find. Okay, there are also intracellular receptors, and those are found within either the cytosol, which is the cytoplasm, or the nucleus of the target cell. They are typically activated by small or hydrophobic chemical messengers that can cross the membrane. So they will be able to cross the membrane and activate the protein within the cell. So examples of hydrophobic messengers would include both the steroid and thyroid hormones of animals, and an activated complex can act as a transcription factor. Transcription refers to transcription and translation, so when we're talking about protein synthesis, so a transcription factor will turn on a gene. So here we have a hormone like testosterone that is able to diffuse through the cell membrane. It binds with this hormone receptor complex right here. And then that actually enters the nucleus, as you can see, and alters the DNA, acting as a transcription factor so that a particular protein is produced. The second part is transduction. So signal transduction usually requires multiple steps, and multi-step pathways can end up amplifying a signal. So there can be very few molecules that can produce a very large cellular response. And multi-step pathways provide more opportunities for coordination and regulation of cellular response. So the molecules that relay a signal from the receptor to the response are going to be mostly proteins. These proteins act like dominoes, so one receptor is going to activate another protein, which activates another protein, which activates another, until there's finally some type of response is produced. So at each step, the signal is what we call transduced into a different form, and this is usually a shape in the change in a protein. So in a lot of pathways, the signal is transmitted by a cascade of protein phosphorylations. So we have these proteins, this group of proteins called protein kinases, and they transfer phosphates from ATP to proteins, which is again a form of phosphorylation. And these protein phosphatases, remember it ends in aces, so that's an enzyme, remove the phosphates from the proteins, and they dephosphorylate the protein then. So this phosphorylation and dephosphorylation acts as a molecular switch, which will turn activities on or off, or amplify them, or inhibit them as required. So here we have our ligand, or our signaling molecule, activating the receptor. So as you can see, there's an activated relay molecule here, and then we have this multi-step process that involves this phosphorylation and dephosphorylation of ATP here, until finally we have some type of protein that has been activated and causes a cellular response. So this right here is called the phosphorylation cascade. The ligand that binds to receptor is always referred to as the first messenger, and the next part is going to be the second messenger. So second messengers are always going to be small. They're non-protein. They are water soluble, or sometimes they're even ions that are spread throughout a cell by diffusion. And these second messengers participate in pathways initiated by GPCRs, which is important. And one of them is cyclic AMP, also calcium ions. These are very common second messengers. So you have your ligand and then your second messenger. So cyclic AMP is one of the most widely used second messengers. And we have adenylyl cyclase, which is an enzyme that's going to convert ATP to CAMP, or cyclic AMP, in response to some type of extracellular signal. So here is what this looks like. So as you can see, we have our molecule here, then our adenylyl cyclase, which forms cyclic AMP here from ATP. Well, I guess the arrow should go the other way. Forms this from ATP. And then with the addition of water and another enzyme called phosphodiesterase, this converts cyclic AMP to AMP. There are lots of different signal molecules that contribute to the formation of cyclic AMP. Other components are G proteins, or G protein coupled receptors, or protein kinases.